This is part 2 of our chapter covering the lymphatic and immune system. In part 1, we talked about some of the structures and organs and tissues and things associated with the lymphatic system, and now for the remainder of this chapter, we'll turn our attention to immunity. So the immune system, which again is kind of hard to talk about because it involves so many different organ systems and aspects. Before we get too far into it, there's one quick distinction that must be made, and that is the distinction between what is known as innate immunity and adaptive immunity. Immunity refers to protecting ourselves from pathogens, things that can make us sick. And when you think about all the ways that the body does this, you can group it into these two categories, which we call the innate immunity and the adaptive immunity. And to understand the difference between these two sides of the immune system, I usually like to think about a castle. So in the same way that a castle has kind of some built-in defenses into it, for example, this castle is on a hill, right? So it would make it tricky for invaders to get into the castle because they got to climb up the hill to get to it. There's also all of these trees that are in the way, which make it hard for invaders to get into the castle. And then once they've made it up the hill and through the trees, then there's these big walls that they would have to scale. And maybe there's a boiling pit of oil somewhere and little traps in different places of the castle to make it hard for the invaders. And then only after, making it through all of those defenses would then the invader get into the castle, at which point it would be facing combat with the warriors or the soldiers in this particular castle. Well, that's kind of a simple illustration as to what the innate and adaptive immune system are. The innate immune system is like the category of things in our body which would be analogous to the trees or the walls or the traps that are set. They're mechanisms that keep pathogens out of our body, but aren't necessarily able to identify and selectively attack the pathogens. And so generally, the adaptive immune system, on the other hand, is going to be characterized by the fact that it's very, very, very specific. The warriors of our adaptive immune system are the lymphocytes, and the lymphocytes can attack and recognize pathogens very specifically in a very effective way. So a little bit of an interesting analogy there. So we'll turn our attention to innate immunity in this lecture, and then in the following lecture we'll talk about the adaptive immune system. A couple of things. The innate immune system is present from birth. So basically all of the components of your innate immune system, they don't get better, they don't develop further during age, they don't become increasingly efficient. You're basically born with all of the equipment that you're going to have in the innate immune response. And also it's considered non-specific. Now this gets a little bit tricky. We'll talk a little bit more about that in a minute, but basically the innate immune system can't recognize specific viruses or bacteria. It's just kind of defenses that are used against all pathogens of all kinds for the most part. Okay, the things that are involved in the innate immune system. These are things like barrier defenses. This would be like analogous to these walls of the castle that help protect it from invaders. Also, there are different cells that are involved in the innate immune system we'll talk about in a minute. There's a kind of variety of proteins which can travel in the plasma of your blood and help to keep you safe from illness, so we'll talk about those. And then there is this process known as inflammation, which will also be considered part of the innate immune system. Okay, to begin, we'll talk about the barrier defenses. There's a nice table in your book that kind of lists a variety of these barriers, and some of them are just simply a physical barrier. Other times they're a physical barrier or also perhaps a chemical barrier, so keep that in mind. The first one that is mentioned is the skin. And this specifically is going to be the epidermis of your skin, so the stratified squamous epithelium. And what's interesting is that the cells in that layer of your skin are very, very tightly packed together, so they don't allow a lot of things through. Also, those cells have a protein called keratin that they make, which almost is like it's almost like plastic in its composition. And it helps to waterproof the skin, so it helps to repel water. Also, what's interesting is that there are Langerhans cells in your epidermis. A Langerhans cell is a special kind of cell, specifically a special kind of dendritic cell that can eat bacteria or pathogens that get into the epidermis. There's kind of an interesting picture here that I was able to find where they were able to stain the dendritic cells. So that's these bigger cells that are kind of sprawled throughout the stratified squamous epithelium of the epidermis. And these are just cells that are kind of standing by waiting in case, you know, a bacterium somehow is able to get its way through the layers of the epidermis. It will come in contact with one of these cells, which will eat it through the process known as phagocytosis. On your skin as well, there is sweat glands and sebaceous glands, which will release sweat, and sebum. Sebum helps to keep your skin supple so it doesn't crack. You know, as soon as it starts to crack, it makes it easier for pathogens to get in. And then also the sweat glands are interesting because not only do they help to wash pathogens off of your skin, they also create a little bit of acidity on the skin, so there's a little bit of a low pH that can be formed on the surface of the skin to help inhibit bacterial growth. And so these are more of a chemical barrier than a physical barrier, which are also effective. In your mouth, there are the salivary glands, and the salivary glands will actually, you know, this is what makes your saliva. The salivary glands will also secrete a enzyme known as lysozyme, which will break apart cellular fragments, such as, you know, bacteria. In your stomach, there's the GI tract, which creates a whole bunch of acid through a pretty intensive feedback loop, which we'll talk a little bit about later in the course. But that also creates a low pH, which makes it more of a hostile environment for pathogens. Then there are the mucosal surfaces. Remember, a mucosal surface is one that is an epithelium with a mucus layer on top of it. And these are going to be made of non-keratinized epithelial cells. And generally they're in areas where there needs to be absorption and secretion that happens. So there can't be many, many layers of dead keratinized cells like there is on the epidermis. One of the more notable mucosas in the body that I like to talk about is the pseudostratified ciliated columnar epithelium in the trachea. Here's a picture of that. And just recall that there are all of these cilia on here, and they create what's known as the ciliary elevator because they're continually pushing the mucus and the pathogens and particles and things that have been trapped. They're continually moving it upward, up the trachea and out of the body. So it's kind of an impressive feat there of that mucosa. Okay, and then also what's interesting is that there is bacteria that are in your gut, in your colon, that are not pathogenic. They don't make you sick. This would be called what's known as the normal flora. A pretty mind-boggling statistic here is that for every cell in your body that's your body's cell, there are 10 bacteria. So on a cell-to-cell count, there are actually more bacteria that make up your body than you. You're more bacteria than you. Of course, a bacterium is much smaller than a normal cell. So even if there's 10 times, 10 bacterial cells for every normal human eukaryotic cell, it still ends up being a pretty small percentage of the volume. But in either case, these will actually outcompete the mucosal tissues in your GI tract, and they'll actually prevent pathogens from growing on the mucosal surfaces by outcompeting them. All right, there are also a variety of cells in your body that help assist in your immunity, but they're not super specific. A couple examples is the class of the phagocytes. Phagocytes are any cell that can consume a pathogen. And there are a couple of examples here. One is the neutrophil. We've talked about these before. These are one of the 5 leukocytes that we've mentioned. There's also the macrophages, and macrophages are a little bit more of an interesting cell because they're a lot bigger. They're more kind of like an amoeba in a sense. So this is showing a really big macrophage, and actually what you're seeing here is little bacterial cells inside of it. So it's consumed. So these are all kind of bacterial cells surrounding it. And this thing is really big when you consider the nucleus and how big all of this cytoplasm is. And it can move around with these little cellular extensions called pseudopods and make its way throughout the body. Macrophages can either roam around throughout the tissues, they're actually kind of mobile, or they can remain fixed in a tissue, either one. There's also the dendritic cells, and dendritic cells are kind of a specialized version of a macrophage. Here's a really cool picture of one, and they're interesting because they have these really sprawled out branches coming off of the kind of the cell body, which means that it's a very far-reaching kind of meshwork of little extensions that can catch any sort of pathogen that makes its way through this particular region. Dendritic cells have different names depending on where in the body they're found. For example, if a dendritic cell was in the liver, we would call it a Kupffer cell. If it was in the epidermis, we'd call it a Langerhans cell, like I mentioned earlier. So there's a variety of these. This particular image is of a fixed follicular dendritic cell. It's anchored itself in a lymph node, and so all of the lymph would be passing through as it's clung to the reticular fibers in a lymph node, and it just kind of hangs out there almost like a spider in its spider web. The macrophages in the dendritic cells, they all come from monocytes, is kind of their lineage. Remember, the monocytes were a type of white blood cell, and they can differentiate to form macrophages or dendritic cells. Another important cell is actually a lymphocyte, and it's a natural killer cell, is what it's called, or an NK cell. And these cells are still a bit mysterious. It's hard to find really good concrete information about how these are unique or how they're characterized different. So if you find any of that information, do send it along. In previous textbooks I've read, the natural killer cells were involved in a process known as immune surveillance, where they would kind of ensure that there was no tumor cells escaping and metastasizing throughout the body. But this textbook and other sources that I'm reading that are a little bit more current suggest the natural killer cell is going to basically just induce apoptosis, not only on cancer cells, but any cell that's infected with a virus or a bacterium. Apoptosis, recall, is a programmed cell death. So it's a way of telling a cell that it should turn itself off and die. This is done by a couple of different ways, and if that's the route, it will use these two weapons here. Perforin is a protein that can be released through a granule, and it can poke holes in the membrane of a cell and then disrupt its permeability so that it dies. And then granzyme goes in and just kind of starts digesting things apart. Now, one of the things that's kind of interesting is that neutrophils, macrophages, and dendritic cells all phagocytize pathogens. But you might ask yourself, well, wait a minute. How does a neutrophil, macrophage, or dendritic cell find the thing that it's supposed to eat, and how does it recognize the thing that it's supposed to eat? Because remember, cells don't have eyes, ears, or brains, so it's harder to kind of recognize stuff. And furthermore, you might say, wait, I thought the innate immune system was the part of the immune system in the body that doesn't recognize specifically pathogens. And here's kind of where the big exception is. On these cells, there's what's known as a pattern recognition receptor. And pattern recognition receptors are a pretty general receptor, and they bind generally these things which are known as pathogen-associated molecular patterns, or PAMPs. PAMPs are basically just characteristic features that you would find in, like, let's say, many different kinds of bacteria or many different kinds of virus. For example, maybe the flagella on all bacteria always has a certain molecule on it. That would be called a PAMP, because it's just a general pattern of a substance that's only found on bacteria, not on eukaryotic cells. Well, there's a variety of different molecules out there which are called PAMPs, and those are bound by the PRRs that are found on these cells. So yes, neutrophils, macrophages, dendritic cells can recognize these molecular patterns that are generally found on pathogens, but it's different in the sense that there's not really billions and billions and billions of different kinds of these receptors that can bind specifically to billions and billions of different kinds of bacteria and viruses. It's just one kind of general receptor that binds general types of things that are usually on pathogens. So that's the caveat. Okay, the next thing that we'll talk about is the soluble mediators. These are proteins found inside the plasma that help fight the good fight against pathogens. The first two words we have here are the cytokines and the chemokines. These are general names used to describe a class of compounds. Cytokines are a variety of different molecules that are used to facilitate an immune response. So if a cell was trying to turn on another cell, or if a cell was trying to communicate to another cell that it was sick or it was infected, it would release a variety of these different chemicals. We call any chemical that's released by a cell to help mediate an immune response, we call that chemical a cytokine. Chemokines are kind of the same idea, but they have the special emphasis that these are involved in chemotaxis. In other words, recruiting immune cells to a particular place in the body. Chemotaxis, again, the way that it works is these chemokines would be released at a certain site in the body, and then the chemokines would kind of spread out throughout the body and get a little bit more dilute. And then basically a cell that is able to find a place through chemotaxis will be able to locate the place of the highest concentration of chemokines. So that would be called positive chemotaxis. It's going up and up and up, forever up the concentration gradient until it finds the source of the chemokines. Another class of soluble mediators are what are known as the early induced proteins. These are proteins that are made and released very quickly after intracellular infection with a bacterium or a virus. Some examples are a class known as the interferons. There are a lot of different interferons, but generally what we think of when we think of an interferon is a substance that's released by a cell that's infected with a virus. So let's imagine we're in kind of some tissue of the body, and here's the infected cell right here. Well, when the cell is infected, this is called an early induced protein, because before the virus has had a chance to hijack the cell and start cranking out new virus, the infected cell sends out kind of like a messenger to other nearby cells. The class of proteins that are released here, or the class of molecules that are released, would be the interferons. There's interferon alpha, interferon beta, interferon gamma. There's a lot of different interferons. But the most notable thing that they would do is go to those neighboring cells and tell them to start producing antiviral proteins. Basically, an infected cell would send out interferons to tell the cells nearby, hey, I'm infected, protect yourself. So it's a general class of interferons. There are also these two here that the book mentions, mannose binding protein and C-reactive protein. Both of these proteins will bind to bacterial cell walls, and they will actually kind of bind to those bacterial cell walls and tell the phagocytes, hey, there's something you can bind to. The phagocytes have receptors that can attach to these two things, the C-reactive protein and mannose binding protein. And so if these are covering a bacterium, then the phagocytes have an easier chance of grabbing onto the bacterium and getting close enough to it to phagocytize the bacterium. This process, generally speaking, is called opsonization, which literally means to prepare to eat. It's just the process of making a pathogen more eatable. It makes it more easily grabbed by a phagocyte. Okay, and then there's the complement system, and this is a pretty involved series of proteins made by the liver. There's about 30 different kinds of these proteins made, and then once they're made in the liver, they diffuse into the plasma, where they will just kind of reside. And there's a wide variety of responses here from this system. Some of the effects of activating this system, the complement system, is opsonization, so we just mentioned that here. Also chemotaxis and perforation. So let me kind of just walk you through this process. It usually begins when an antibody, in this case we have kind of two antibodies, attaches to some specific antigen on a invading pathogen. There are a couple different pathways by which this complement system can work. One is shown here. This would be considered what's known as the alternative pathway, and that is the pathway that is employed by the innate immune system of our bodies. There's a pathway called the classical pathway, which is utilized by the adaptive immune system. So keep that in mind. We'll just look at the alternative pathway here. All right, so once the antibodies have bound to the antigen, then what happens is that there's going to be a split that happens. These two subunits will split into forming what's known as C2 and C4. A chunk of C2 and a chunk of C4 will come together, and that will form what's known as the C3 convertase, and that will be used later to convert C3, which is another protein in this system, into two little bits, C3a and C3b. Now here's where we kind of start to see the wide-reaching effects of this activating this system. C3a attracts phagocytes, so in that sense this is kind of where the chemotaxis part of the system is employed, because this is going to recruit phagocytes. And C3b is going to actually bind to the surface of pathogens, rendering them more easily phagocytized. So that would be the opsonization facet of this system. So complement helps to provide chemotaxis. It also provides opsonization. And then the third is that it perforates the membrane of the pathogen. So here's where we continue going on further. One of the fragments from C3, so one of these here, either C3A or C3B, I don't know if it matters which is used, will bind back to C3, and that will form what's known as C5. And C5 is going to split and form C5A and C5B. One of these is the active form, C5a. C5a is what actually inserts and attaches to the membrane of a pathogen, and as soon as that attaches, it recruits a whole bunch of other proteins into it to form kind of a channel in the membrane of the pathogen. And once a channel is made, then there's no longer selective permeability. Stuff starts leaking out of the cell, and then it's destroyed. All right, so this is called this whole complex of complement that form a whole is called the membrane attack complex, or the MAC. And that would be the third way that complement is utilized to destroy pathogens. Now, if this was an immunology class, I might have you learn this whole pathway. I had to learn it back in the day. But because it's an A&P class, we'll say, yay, you don't have to worry about this. But I do want you to know and kind of see how this one pathway yields these three different results. Okay, one of the last things that's employed by the innate immune system is inflammation. And inflammation works something like this. It starts off with tissue injury. And in connective tissues throughout the body, there are these cells called mast cells. And mast cells are super good at starting inflammation. A mast cell is kind of like a basophil inside the blood, but it's not inside the blood. It's out in the tissues, in the connective tissues. And so when there's injury to the body, generally what will happen is that the mast cells will start releasing histamine, and histamine is the big compound that initiates the inflammatory response. Parts of this response involve vasodilation, and with vasodilation there's also an increased vascular permeability. So not only are the vessels here getting wider, they're also becoming more permeable. The vasodilation will cause more blood flow to the area, which is kind of why places that have inflammation appear more red, and also are usually a little bit more warm to the touch because they have more warm blood going to those regions because of the vasodilation. But then also because of the endothelium is more permeable, the vessels are more permeable, it allows neutrophils and things to get out a little easier. So there'll be kind of more fluid being pushed into this area. There's going to be more white blood cells getting to this area. The fluid that's accumulating could be fluid that's just seeped out of the vessel because it's more permeable. That can cause kind of the swelling that you see in inflammation. And so altogether there's warmth, there's generally swelling, there's usually redness, and there can sometimes be pain. And ultimately it allows for the recruitment of phagocytes and other white blood cells into the site of injury. That's going to wrap us up here just for the innate immune system. Stay tuned for the final showdown where we talk about the adaptive immune response.